So the logic used by the sequest algorithm to search experimental spectra in the already present databases is just based on a linear search. So as we know that these databases comprises of the proteins that are canonical, and they are different from the proteins that are actually present in the cell. And as we know that in the cell, the proteins that are being translated from the messenger RNA, they also undergo various modifications, and that are very important for their proper functioning and for their regulation. And these modifications are either permanent or reversible. And we also know that the most of the proteins, maybe more than 90%, they undergoes these modifications. So for example, as we have discussed the modification that the proteins they undergo, and the example is the enzymatic activity of some proteins is regulated by the addition or the removal of a phosphate group. And this removal of phosphate group is at a very specific residues on the protein. So phosphorylation is a reversible process, and we know that the protein kinase, it can add the phosphate groups, and we know that whenever the kinase add the phosphate group in the protein becomes active. And we know the many examples of these proteins, they are present in the glycolysis. And the other protein and the enzyme is called the phosphatases that remove the phosphate groups. So these are the modifications that are being going on in the cell. So in the cell, proteins form a complex system, and they undergo modifications as we discussed earlier in the cell, and that is very useful for their functioning in the cell, and they perform very important function like cellular signaling and the metabolism of the cell. And these proteins, if they do not undergo the modifications, many important cellular functions cannot be performed by those proteins. And the most important modifications that proteins undertake are the glycosylation and the phosphorylation. And we have already seen the example of phosphorylation kinase and phosphatase enzyme. They perform important role in the phosphorylation process. And as we know that most of the proteins, they undergo the modifications within the cell. And I told you that these modifications, they are very important to perform the proper functioning in the cell. And these modifications may be more than 200 of different types. And these includes the phosphorylation, glycosylation, acetylation, and many others modifications. So when the protein is formed, I means that when the protein is translated from the messenger RNA, these modifications, they are undergone in the cytoplasm, generally in the ribosomes and in the Golgi complex. So after these modifications, the functioning of the proteins, they are performed in a very perfect way. As we know that these modifications, they are important, and we cannot find the modifications at the DNA level. So we have to find the modification in the naturally occurring proteins. So computationally, a chemical modification of a protein, that is P1, P2 up to the Pn at position I, results in increased mass of the N-terminal peptides, that is Pi plus 1 up to Pn, and increased mass of the C-terminal peptides. As you know, there are two terminals in the protein. One is the N-terminal and the other is the C-terminal. N is the amino group, C the carboxylic group N-terminal. And the increased mass of the C-terminal peptides, that will be the P-1, P-2, minus means the charged particle ions, and up to the Pi-1.